So today's topic, just we are going to speak regarding to a species, that is zebrafish. So I hope here 9 students will be there, same as the students who have passed the third form D as well as M form peoples will be present in the seminar. So this species, zebrafish, whether it is known to you or not, I don't know. But this zebrafish, it has been emerging as like anything, as a species work in different pharmacological studies, in different pharmacological studies. The zebrafish, it is called as nellore. It is present in a tropical freshwater fish, belongs to the family Cyprinidae. It is a proper aquarium fish. It is frequently sold under the trade name of zebra danio. It is an important vertebrate model organism in a scientific research. So this is a basic introduction what I want to speak regarding to this particular species name called as zebrafish. Students as well as the participants, you may be hearing, if it is a pharmacology lab in animal house, you may see different type of species like mice, rat, hamster, guinea pig, rabbit, right? As like that, you may have studied in books in practical section, second part of the practical in third year or fourth year, you will be studying this particular thing. Even about this dogs, cats, monkeys, how they are being used in pharmacology lab. Am I right or wrong? So, but you may not be aware of this particular species regarding to this particular zebrafish. So, as per CPCSA right now, they have given permission as well as they had made many pharmacology peoples to make awareness regarding to this particular species throughout this world regarding to this particular species, how it is playing a role regarding to this particular pharmacological studies. So that's the initiation of me right now. It is making me to stand before you right now, right? So followed with that slide, the zebrafish has become an important vertebrate model organism for studying this disease and for assessing the efficacy and toxicity. And it has emerged as an ideal complementary model system for the drug discovery. For your kind information, it is well suited for genetics, pharmacological studies, toxicological studies, as well as kinetic parameters. It is sharing nearly around 60 to 80% of homology with humans and exhibits unique characteristics such as short generation time, large number of progeny per cross and transparency, extrauterine development of embryos, and rapid organogenesis. The main use of this particular zebrafish in drug discovery and development is a target validation as well as lead optimization. In addition with that, drug toxicity and drug repurposing studies. So this was being focused mainly in the research, drug discovery as well as development. So the main target is target validation, lead optimization as well as drug toxicity and drug repurposing studies. Combinatory chemistry combined with this high through and ultra high through generates the large pool of preliminary heights. Validating these heights by mammalian animal model is expensive and very slow, resulting in a gap in a drug development process. That's the reason why the zebrafish has been taken as a model, due to because of long duration as well as cost, economical. Due to because of this reason only zebrafish has been emerging as like anything, right? The zebrafish can fill the gap in a preclinical pipeline between in vitro assays and expensive screening using mammalians. What I have said earlier. Further, to increase the success rate, it is extremely important to test compounds using whole organism vertebrate models during early stages of drug development. Why this is called as a zebrafish? Why it was being called as a zebrafish? You can able to see the fish which is located on the screen. There's a strip. You can able to see the blue strips. It comes from the horizontal. It is laying on horizontal, right? So horizontal blue strips on each side of the body. That's why we are calling it as zebrafish. Even zebras, they will be having some strips. Yeah, that's the reason why we are calling this particular species as a zebrafish. It is named due to their strips prefer to live in large groups called as shoals. So many group of animals in a place it can able to live, it can able to survey. That's a reason they are being called as this particular species, zebrafish. Coming towards the taxonomy. Species, what we are calling it as Rio. Genus, what we are calling it as Danio. Family, what we are calling it as Cyprinidae. Order, Cypriniformes, and the class of Actinopterygii. And this is about the taxonomy. And coming towards the general features regarding to this appearance, habitat, feeding as well as reproduction. Coming towards appearance part, the dimension is nearly around 4 centimeters. Silent distinguishable features of male and female. Often transparent adult bodies will be present. This is about the appearance. Dimension is nearly around 4 centimeters. Distinguishable features of male and female. Often transparent adult bodies will be present. What is the benefit? What is the benefit of this particular thing? Means a large number can kept easily and cheaply in the labs. For example, if we are using mice or rat, only in a cage we can able to use 6 animals, but in a bowl we can able to maintain 50 zebrafish, right? So that's the reason what we are calling here. Large number can kept easily in a cheaply in labs. Good model for visualization of cellular activity. So this is a valuable point what I want to make here. Cellular activity. So this particular point. Habitat. It can able to be live in fresh water as well as tropical fish also. And the availability regarding to this particular species is universal. And regarding to this particular feeding, it is coming under the category of omnivores. And the maintenance regarding to this particular species is very low cost. In reproduction, female spans every 2 to 3 days, breeds all year round. Several hundreds of eggs it can able to produce in a single clutch. External fertilization. So regarding to this is about the reproduction. Regarding to the benefits, large number of offspring, good for batch variation studies. So at that time we can able to get a more number of content, that is regarding to zebrafish. and easy availability of that particular eggs. So these are the major things what I want to quote regarding to the general features, general features of this particular species name called as zebrafish. And coming towards the distribution, the zebrafish is a nature to the streams of the southeastern Himalayan region. So the native of this particular zebrafish is southeastern Himalayan region. Also, in eastern India, it is also native towards Pakistan, Bangladesh, Nepal, as well as Myanmar. It commonly inhabits streams, canals, ditches, ponds, and slow-moving to stagnant water bodies, including rice fields also. Zebrafish have been introduced in aquariums in Japan, Canada, United States, as well as Australia. So this is about the distribution of that particular species. And here you are seeing the anatomy of the species name called as zebrafish. Just for your information, and this is regarding to the development of the stages involved in zebrafish, starting from zygote, ending towards adult. It takes nearly around 3 months to be an adult. So within a 15 minutes, it can able to form a zygote. Within 45 minutes, it can able to form a cleavage, as well as gastrulae in 2 hours. In 2 hours, it will be playing a role. Segmentation within 8 hours. Hatching within 16 hours. As well as in a larval stage, it takes 48 hours. And within 3 months, it can able to be a perfect 100% adult zebrafish. So this slide is representing regarding to the development stages of zebrafish. And this is a picture which is representing regarding to the larvae. You can able to see here, the species was very transparent. That's the reason why we said in the initial slides, in cellular studies, and the mechanism what was happening inside, it can able to be visibly seen macroscopically. If it was seen in microscopical, it is well and good. It is well and good. But even in macroscopical, we can able to identify, or we can able to analyze what was happening, right? So this is a larvae. What is regarding to this fish name called as zebrafish. And coming towards the advantages regarding to the species name called as zebrafish, optically translucent embryos, rapid hatching of eggs. The maintenance cost is significantly lower than those of mammalians, what we are using in pharmacology lab. Amenable for more cellular and genetic analysis. As a zebrafish eggs are fertilized and develop outside the mother body, it is an ideal model organism for studying early development, So these are the things what we are identified and what researchers have identified as the greater advantages of this particular species. And coming towards the disadvantage regarding to the species, several mammalian organs are not present in the zebrafish. So that's a minus point. Although whatever the thing, in mammalians, the organs which was present, it was not present 100% in this particular zebrafish. For example, like breast tissue, lungs, prostate, followed with the skin lacks some specific cellular components found in humans. Skin lacks some specific cellular components or compounds found in this particular humans. Ectothermic physiology was not identical towards this particular humans. Physiology was not identical towards this particular humans. But if you are seeing this particular mice or this particular rat, the physiology is same as it is. like a human. That's the reason why we are using this mice, rat, or this particular monkey in our clinical trials, other preclinical trials. These fish are greatly influenced by their environment and many environment variables, including temperature, lighting, population density, water quality, and nutrition, must be tightly controlled in order to accurately interpret the data which was being obtained. So these are the common things what the researchers are saying it has an disadvantage. So first point, it is saying the mammalian organ, it is not accurately present in this particular zebrafish. Same as like that, comparing to humans, the physiology is not equal with this particular species, right? Then followed with regarding to environmental influence. So these are the three things what has been considered as a disadvantages regarding to this particular species. Next, we are moving towards the topic. Why it is considered as a model? Why the zebrafish is considered as a model for a human disease? Why? Because nearly around 70% of the human genes are being found in this particular zebrafish. That is a main reason why this is considered as a model for human disease. I quoted very clearly here, 70% of human genes are found in this particular zebrafish. That is a main reason. In addition with it, the zebrafish is having two eyes, a mouth, brain, spinal cord, intestine, pancreas, liver, bile ducts, kidney, esophagus, heart, ear, nose, muscle, blood, bone, cartilage, and teeth. So these are the parts which was being present on this particular zebrafish. Many of the genes and critical pathways that are required to grow these features are highly conserved between humans as well as this particular zebrafish. Thus, any type of disease that cause changes in these body parts in humans could theoretically be modeled in this particular zebrafish. So that's the reason why we have picked this particular species, why this scientists have picked this particular species as a model. Why to use this particular zebrafish when there is a species name called as mice? So already there is a species which was being used for many clinical trials, preclinical trials. Especially for a basic preclinical trials, we will be using this particular mice as well as rats. So now the CPCC is recommending, instead of this particular mice as well as rat, if possible, you can able to go with this particular zebrafish. So for that particular reason only, I'm making this particular slide right now. While mice are evidently more similar to humans because they are mammalians, zebrafish have several advantages over their furry competitors. Whether I'm audible?
Yes, sir. Yes, sir.
One important advantage of the zebrafish is that the adults are small and prefer to be housed in large groups or shoals. Because of this result, they require much less spaces and they are cheaper to maintain that particular, maintain comparing towards this particular mice. The NIH zebrafish core houses hundreds of thousands of zebrafish in a state-of-the-art facility. Another advantage is that the adult zebrafish breed readily and can produce as many as 50 to 300 eggs at their time. This is quite different from mice as they generally produce litters of 1 to 10 pups. So that is the main advantage. What I want to speak here, Dash. So in a cycle, it can able to yield more number of eggs. So that's the valuable point what I want to make. But if it is in mice or rat, you can able to see not more than 10 it can able to yield. But you can able to see here approximately 300 eggs are being in a cycle it was being producing. Minimum 200 able to be no chance of destruction or otherwise destroying regarding to this particular species. So that's the reason we are using this particular mice as an animal model, comparing towards this particular mice. And coming towards differentiation in sex regarding to that particular fish. Males are a bit smaller and more slender than females. Male zebras appear more torpedo shaped, while females tend to have a larger belly. You can able to both sexes have two pairs of strips, but females are usually larger comparing towards male. More full-bodied than the particular males, a female's belly balloon when it fills with that particular eggs. You can able to see like a balloon in female zebrafish when it was carrying this particular eggs. And coming towards the benefits regarding the zebrafish. The zebrafish is small and robust. They are cheaper to maintain than mice. The major important what I want to speak regarding to zebrafish here is many of the species you can able to see only in the dark time you can able to yield the eggs. You can able to lay the eggs. But the zebrafish in day and night you can able to lay the eggs. Even the mating can able to be done in day as well as in night. Many species mating will be done only on night time. For the zebrafish, there is no limitations regarding to that particular thing. Zebrafish produces hundreds of offspring at weekly intervals itself. That's a major thing. That's a major benefit also. They grow at an extremely fast rate, developing as much in a day as human embryo develops in a month. So these are the things what we are considering as a benefits. In addition with that, zebrafish embryos are nearly transparent, which allows the researchers to easily examine the development of internal structure. What I have said earlier, and I'm showing this larva. So I said it is transparent. So it is very easy for the researchers to examine for the development of interval studies, right? Every blood vessel in the living zebrafish embryo can be seen using just a low power microscope. That's a great thing, right? So but in mice or rat or any other species what we are using, so it is not much visible, not much visible. So the blood vessels in living zebrafish embryo, they can able to be seen using just a low power microscope. Zebrafish have a similar genetic structure to humans. Already what we have said, nearly around 70% of genes are quite comparable with the humans. 84% of genes known to be associated with human disease. Make a note very clearly here, nearly around 84% of genes known to be associated with human disease have a zebrafish counterpart. In addition with that, as a vertebrate, the zebrafish has the same major organs and tissues as the humans. There is a slight difference, that's it. The zebrafish have the unique ability to repair the heart muscle. This is a great thing what now the research is moving on right now. Zebrafish has the unique ability. No other species is having this particular ability. That heart muscle will be repaired by their own mechanisms without any external effect. But the study was not being properly studied, but the study was going under that particular thing. How the repair of heart muscle was being going on. A part of their heart is removed. They can grow it back in a matter of weeks. So see, within a week, it can able to grow its own muscle, especially this particular heart muscle. That's the reason the scientists are working to find out the specific factors. What is the reason for it? Which is involved in this process to see if this will help us to develop the ways of repairing the heart in humans with heart failure or who have suffering regarding to attacks of this heart. The zebrafish genome has been fully sequenced to a very high quality. This has enabled scientists to create mutations in more than 14,000 genes to study their functions. See, these are the great things what was happening regarding to this particular zebrafish. And coming towards the major area of today's section, regarding to this drug discovery, how this particular zebrafish was being playing a role and where it was being emerging. In the last decade, the technology has been extended to the generation of high value knowledge on safety risk of novel drugs. The larval zebrafish appear to combine the advantage of whole organism phenotypic assays and those of in vitro high throughput screening techniques. The zebrafish is used to identify the potential safety liability, particularly before lead candidate selection. The zebrafish was being used in the drug discovery regarding to cardiovascular system. In cardiovascular system, it is used to reveal the decrease in heart rate as well as atrial ventricular dissociation. In central nervous system, behavioral assays have been and are further being developed into screening platforms for assessment of locomotor activity. Example like convulsant, conjunctive impairment, drug dependence potency, impaired visual as well as auditory functions. In those areas and all, the studies was being going on. In addition with that, bone density, GIT function, renal function, right, as well as this toxicological studies. So these are the areas where this particular zebrafish was being implemented for a drug discovery. Once again, cardiovascular, central nervous system, bone density, GIT, renal system, as well as in toxicological studies, this particular species was being used majorly. And coming towards the basic thing, the in vivo pharmacological studies in zebrafish. So how to select? You can able to see different colors of zebrafish in the slide right now. So among this, they are all considered as a zebrafish, especially the blue strips as well as the silver strips. They are being considered mainly as a zebrafish. Even there is a gray strain, yellow strain, pink strain, red strain, right? But in research, they will be using the silver as well as blue strips, right? So already I have said this zebrafish will be called as Danio Rio. Even in this aquarium shops, you can able to get these fishes. In research, we will be using an adult fish. If it has crossed 6 months and it is before 8 months, this type of adult fish will be used for the study. And the length of the fish should be nearly around 4 to 5.5 centimeter. Both the genders, what I want to say, sex, can be used, male or female, both can be used. As like this mammalian species like mice, rat, how we are doing toxicological studies. For this species also we are performing toxicological studies, that is acute toxicological studies. As per OECD guidelines 203, these toxicological studies are being performed. Just we will be taking 5 different type of concentration. So in a group, we will be taking 7 fishes. So it depends upon the researcher how many groups they are taking. But in a group, we want to take 7 fish. Final peoples know, as well as many informed peoples also knowing about this. If you are taking mice or rat in a group, n is equal to 6. That is nothing but we want to take number of animals is 6. Why? Because the average. So the outcome will be extraordinary. That's the reason why in mammalians, for example, mice or rat, we are taking 6. In this groups, we are taking 7 as a model for an average. So the animal was being exposed towards that particular drug, which is filled in the tank, nearly around 96 hours. And the mortality death was being recorded for every day, that is 24, 48, 72, 96 hours. Every day for 5 days to 6 days, we are monitoring the mortality of the particular species. And further, as like this particular studies, we are going for LD50, that is concentration which kills 50% of the fishes. So by that, we are taking this particular LD50. So this is the basic study, what we are performing in this particular zebrafish. Same as like that, what I have included in earlier slides. This zebrafish was being assisted in cardiovascular, central nervous system, as well as renal, right? So these are the points. Bone density I have been included. So in this CNS assessment, for example, like memory, learning, neurochemistry, visual, auditory, electrophysiological examination. So these are all the things what we are examining. Behavioral changes, the coordination, motor reflex. So these are the things. In addition with that, the temperature, body temperature. These are all being. These are all being monitored in this central nervous system. Right, fine. So coming towards this behavioral protocols. So you have been aware of this particular activity, I think so, locomotor activity. I don't want to make much more time, just I will make it very short right now. So I hope the final year students as well as PG students, they will be aware of this particular activity named called as locomotor. In our college, sorry, in your college, you may have performed with the help of an instrument name called as, sorry, kindly mute, my dear. Jashwant Kumar, kindly mute. Locomotor. Jashwant Kumar, please kindly mute your phone, please. Yeah. So locomotor activity. In this locomotor activity, in your college, you may have performed earlier. You have seen the instrument name called as locomotor. You can able to see there will be a photo cells. So you will be placing the animal, that mice, and you will be recording when it is crossing that particular photo cells, right? So that will be recorded. And before adding drug, after adding drug, how many photo cells has been crossed by that particular animal. So this is the thing what you will be recording in this particular locomotor activity, or the motor action. Same as like that in this particular thing also. In this particular thing also, just we will be taking a tank and we will be drawing a line. Just we will be fixing the camera. In this camera, we will be monitoring very clearly. Just after adding the drug and before adding the drug, how many lines it has been crossed. For example, tank is transparent, am I right or wrong? So we will be drawing a line like square or horizontal or whatever the thing, it depends upon us actually, right? So up, down, as well as side, left as well as right side. So just we will be monitoring with the help of a camera also and with the help of a macroscopy also, how many lines this particular animal is crossing before and after adding. So this is the thing what we are performing regarding to this particular locomotor activity. Okay, so just that particular what it is tank sizes 30 into 15 into 10 actually, right? It's not mandatory that you want to maintain this upon your availability, right? You know, availability, right? Same coming towards next, that is going to see. I want to show this slide. In mammals, in mans, what research has came, and in zebrafish, what research has came. For example, clozapine. For example, clozapine, right? Fluoxetine, melatonin, diazepam, pentobarbital, ethanol. So these are the drugs which has been examined in both male, that is man, as well as zebrafish, but it has given a unique answer. See here, in all the things, in both the species, these drugs have given the same action. So this is about the reference of that particular thing, right? And coming towards next, that is regarding to aggression. That is regarding to aggression. We are able to say when we are anxiety, when we are somewhat, what to say that, when we are angry, right? We shout, okay? Then followed with, we will find a place where we can able to be comfortable. Am I right or wrong? So we will be always isolated when we are angry. Am I right or wrong? When we are angry, we will be finding a place where no one is coming. Am I right or wrong? Same as like that. In males, in mammalians also, for example, in cages, if you are monitoring that, if animal was not comfortable, it will not roam in and around that particular cages. It will be staying at the end of the cages, at the corner of the cages. Why? Because it is scared. It is scared. It is feeling fear. Same as like that. In this also, what we are going to do is we are going to take a tank, but normally you can able to see the fishes. They will not stay in the same place. They will be moving here and there. Am I right or wrong? So it is normal activity. But if it is consuming the drug, if it is anti-anxiety, or it is suppressing the angry, that is depression, for example, antidepressant drugs. If it is as like that, means when it is consuming that particular drug, and just it will be moving towards bottom of the tank, and it will be moving towards the corner of the tank. So that is the end point. That is the thing what we are detecting it as. What we are detecting it as. So this is feeling discomfortable, right? So by this way, we are going to perform this particular aggression. Same as like that memory also. You can able to see in our regular practicals, we will be using this particular T maze, Y maze, star maze, plus maze. With the help of mice, you will be performing. Same as like that, same as like that, same as like that. So you will be making a partition in this particular, what to say that, in this fish tank. You will be keeping the food in that particular specific place. Just you will be making the animal to find, to take that particular food where that particular food was being present. So this is the same style what we are performing in this particular maze or plus maze or Y maze or star maze, right? Same, just this is a learning procedure. Same as it is, just the content is different. Here we are using T maze, here we are using what? That is fish tank. In that particular thing, we are making a partition. That's it, right? Same as like that, we are going to perform this anxiety also, right? So all central nervous system activities can be performed with the help of this particular zebrafish. So that is the thing what I want to conclude with the help of the previous slides. And coming towards next, the zebrafish as a model in different studies, how they are being used. So the most valuable slide, what I want to say, right? Regeneration of heart. Here it was being used as a model. Tail regeneration, retinal regeneration, human pigmentation, cancer research, autism. These are the areas where the zebrafish was being used as a model for the study, right? Generation of the heart muscle, tail regeneration. You can able to see this lizard. If the tail has been cutted, it can able to regenerate. Same as like the tail regeneration, retinal regeneration, human pigmentation, cancer research, as well as autism. These are the areas where this particular zebrafish was being used widely for studying. And coming towards the final slide of today's section, further proposals or prospects where we can able to approach, as a zebrafish are now being used in virtually in all disciplines, from neurogenesis to oncogenesis, from behavior to genetics. Its popularity is steadily increased. So now this particular species usage has been increased throughout the world. The emergence of the zebrafish in conference and the increasing the presence of zebrafish studies at large annual meetings are also boosting the use of zebrafish as an appropriate model of this particular organism. Right nowadays, many webinars, even this what to say that earlier, there will be a seminar, or we will be calling this type of studies as a workshop. How to handle zebrafish, how to produce general anesthetics, how, as well as how to isolate the brain, right? So how to be much comfortable with this particular species to work. Regarding to this type of things, many workshop has been started due to because of this pandemic. Last 1.5 years, the presentation was going on through virtual. So I have taken this particular time duration as an opportunity to introduce about this particular zebrafish to our younger generation, younger scientists who want to work in future regarding to this toxicology or central nervous system or cardiovascular, all those things, right? Thank you all. Thanks for your patience. Till now, what you have listened to me. Any queries, any questions? I'm ready to answer. Thank you. Thank you all. Yes, Mr. Sunil. Sunil, sir?
Sir.
Yes, sir. My session is over. If any query is there or any questions is there, I'm ready to answer.
Okay, madam. Oh, okay, sir.
Yeah.
Madam Sweta, madam. Madam, your mic is mute condition.
Okay, sir. Okay. I think nobody is having any queries. If you have any queries, you can message me. I can ask to share, ma. Hello, anybody? Sir, I think nobody is asking. Okay, sir.
No problem.
Okay. I deem it's a great honor and privilege to propose the vote of thanks for me in this event. First and foremost, I'm apologizing you for between the disturbances, and I'm also thankful for our special guest, Dr. Tamil Jyoti Sergaru, to despite his busy schedule and has found his time for this national webinar and giving such a great, valuable and informative and interesting talk and importance of in vivo pharmacological now in this emerging technology on these zebra fishes, and what are the activities we can do in this we can know and we can do in this pandemic situation also. I hope it is interesting and profitable for all the students. Thanking you very much, sir, for your valuable information. Thanking you all for all the participants for your active participation also. Thank you very much, sir.
Thank you, sir. Thank you, ma'am. Thank you. Thank you very much.
Thank you, sir.
Sunil, sir, once again, and thank you very much for your warm invitation too.
Thank you, sir. Thank you, sir.
Yeah, welcome.
So thank you, sir, for giving a valuable information, sir.
Sure, sir. It's my pleasure. Thank you. Yeah, I'm winding up. Thank you all.
Yes, sir. Thank you, sir.